Males. Oh, okay. Just says recording is in progress. All right. Um, That is, I think that it's possible that the males are finding females. They might be seeing females across the desert during the mating season. You see, scorpions have these beautiful eyes. They actually see 360 degrees. Um, so we could talk about this too, but no one's really tested this idea. I actually wouldn't think they're seeing the green directly. I think they might be seeing almost like a black hole. That is, since the UV is being absorbed and only about 2% is being readmitted as fluorescence, you might be sort of a sink, an energy sink. So the UV would bounce off the sand around the animal, and the female would appear almost as a black silhouette that maybe the animal, the male, could cue in on. So just a crazy idea. If someone needs a thesis project, there you go. I got some scorpions with me. Let me just hold up a couple for you. I got a black light. Let's start with this guy here. This is a male Pararoctinus utahensis. And of course, you got to be a little bit careful when you grab them. And I'm holding the animal's stinger between my fingers there. And otherwise, he's going to nail me. This is a male, just a small, beautiful animal from Western Texas. I'm going to turn the lights off in my room and turn on my black light just so you can see. Let's see how this works. Oh yeah, there we go. Is it going to get it? I may have to or if I have to put it by my face or something. You can't. Let me unblur my. Well, anyway, you get the idea. Sorry, it doesn't come in better. I would have to go out and do an un. It might be actually better with the lights on. Let's see if this is going to look better. Okay, let's try this. Yeah, and you can see how he's bugged by that. When the light hits it, it really reacts, and it's not at all surprising. Sorry, I can't get you. There's a little bit better image there. Okay. All right, so scorpion fluorescence, certainly, hopefully, I think I already saw some socks that are using fluorescent yarn to make some scorpions, which is awesome. Okay, and then just a couple of other things before we get into this navigation piece. Scorpions have incredible sensory structures all over their body. As I mentioned before, they've got eyes, actually really good eyes. Early literature said that they have poor vision. That's not true at all. It's not. They can see in 360. These medial eyes have lenses. They've got little eye spots at the corners here of their prosoma. So they've got basically eight eyes, but it's these medial eyes that are most interesting. And they can see down in starlight level, like 10 to the minus seventh iridians, really low level starlight on a moonless night. So I think they can actually see the bushes all around them. They've got temperature and humidity receptors. They've got these organs on their tarsi, their feet, that detect humidity. And on their claws here, on their pedipalps, there's an organ called a constellation organ, a very understudied organ. They've got mechanosensory hairs all over their body. They've got these trichobothria on their claws. Mechanosensory hairs, all you can see them kind of here on their feet and so forth. But they have these slits and cilia. I do want to just focus on these. On each of their eight legs, they've got these little grooves right there, basically on their ankle joint. And these are called basetarsal slits and cilia. They are vibration detectors. They are really sensitive. I mean, really sensitive. They might be the most sensitive vibration detector on the planet. In fact, they can feel a cricket walking in sand up to a half a meter away, which is remarkable because sand, is unconsolidated. Loose, unconsolidated sand is not supposed to be able to transmit vibrations very well at all. But this is how scorpions hunt. So here comes a scorpion out of its burrow, and she's walking along, and she'll just sort of move around on the sand at night, and then she'll stop and spread out her legs, something like this, until some poor little unsuspecting cricket comes along. And just by the vibrations of that cricket and the vibrations emanating from that cricket and spreading across the 8 legs, the scorpion can figure out the angle and the distance to that cricket, grab that cricket, sting that cricket, and eat the cricket. Essentially, they look like they're smoking the cricket. They will hold the cricket in their chelicerae, their mouth parts, and spit out enzymes, digestive enzymes, and suck in the juices. So for the next couple of hours, they actually look like they're smoking a roach, something like that. So that's how scorpions hunt. Here's one I found. I took a picture of a scorpion under a black light eating its meal of a desert cockroach. And of course, the cockroach doesn't fluoresce like the lovely scorpion. So, slits and cilia are a very important sensory organ, but to me, the most important are the pectins. That's what I've sort of given my life over to. These are, well, first of all, 2 lessons. All scorpions have pectins. That's lesson number 4. And lesson number 5 is that pectins are really cool. So, and they are. So let me try to convince you of how cool they are. They hang down from the bottom of all scorpions. They essentially brush the ground. Most people only carry one comb. I always have 2 combs. And so they're right mid-body here, and they drag along the ground as the animal walks, and they really are like combs. Here is a scorpion. This is a male, Smeringus mesensis from the Mojave. This is a video I took when I was a grad student back in the oh my goodness, I guess this is early 90s. And what I'm going to show you here is a male that's about to wake up. So I put him in a small arena. And over here, I have sprinkled an organic wash from a female. I took some chloroform methanol and dipped her in it, and then I dried down the extract on some sand grains and just sprinkled them over in this part of the arena. And so I want you to watch this animal as he wakes up. So this is a male, and he's going to move around the arena. And I'm setting this to a little bit of Al Green because you'll see the mating dance here. Here it comes. So he just woke up, and he's going to start moving over. He hasn't been over in that part of the arena yet. And now watch what happens right here. He backs up, and look at the pectins sweeping the ground. And he's picking up her scent, her pheromone is what's going on. Uh, and there's a cool behavior that we don't know what this is about, but he starts wagging his tail. He might be releasing something himself there, but you see how the pectins are highly active. Now he moves into this very jerky behavior, and this is called juddery. It's a pre-courtship behavior, and if you're lucky, usually in August and September, and you're out there in the sands of Arizona, you might see males doing this jerky behavior, which is an indication that he's picked up on the pheromone and he's probably sending a vibration signal to her to squelch her cannibalistic ways because these scorpions are cannibals. This particular species is. And so he doesn't want to be dinner tonight, and so he's sort of saying, hey, I've got your signal, and I'm now doing my courtship dance. I would like to mate. Now, something cool is going to happen right here in a second. As he moves towards the middle, which seems to be the hottest part of the pheromone, he's actually going to look like he's going to reach for her. It's as if he's so convinced that she's right in here. Watch this. He'll back up and grab for her right here. Look at that. It's just, I feel a little badly for him, but nonetheless, just such an amazing behavior here. induced by this wash from the female cuticle. So it's an indication that scorpions have pheromones and that the males are using their pectins to detect that signal. When you remove the pectins, they don't do that. So this is a workup of that very paper. It's a 92 paper in Ecology, and you can read it yourself. But it shows the tail wags. Sorry about the tail wags and the judders, those little jerky behaviors, and then the pedipalp reach where he tried to grab for her. Um, so what we think's going on is that if this is, and by the way, this species, they, through most of the year, the males and the females maintain their own separate burrows. But in the mating season, basically late summer, the males will leave the security of their burrow and look for females. So let's say this is a female and she's walking around on the sand around her burrow and she's putting some sort of an apron of pheromone down on the sand so that when a male. Oh, look at that. Here he comes now. He's now wandering across the sand looking for females, and when he comes upon that signal, he starts doing that jerky behavior, that juddering that we mentioned. And we think it's a vibration signal to basically squelch her attack tendency because, she's a cannibal. And by the way, females eat more males than vice versa. If she's willing, she will allow him to approach her and grab her claws, her pedipalps. And then they engage in what's called the promenade à deux, the dance of two. It looks very romantic. It's actually not that romantic. He's dragging her along now. And what's going to happen, mating in scorpions, is that so the male is the one that's leading. And if you're lucky to see this out there in Arizona at night with your black light, if you see a couple engaged in the promenade, just put a little twig or a stick right behind the one that's leading, behind the male. And what will happen usually is that he will stop and survey that rock or twig with his pectins. Where are my pectins? Where are my pectins? He'll taste, he'll start feeling that twig, and then he'll extrude. There's an opening, the genital opening is right mid-body on both the males and females. So he'll extrude a spermatophore, which is a sperm capsule, and it's got a sticky end, which will then stick to the twig or the rock, and it's got a little trigger on the other side. And once he's done that, he will carefully maneuver her over the top of that spermatophore, and that trigger will hit her genital opening and shoot the sperm inside of her. And so fertilization occurs internally, and the eggs hatch out inside the mom, and 12 months later, the babies crawl out. So there's basically a 12-month pregnancy in which the little embryos feed off of the juices of the mother. We'll let this couple get on with their life. And then, as I say, they will then split up because the female might eat the male still, so he gets away. But 12 months later, the babies crawl out. And here's a great picture of one of our local scorpions, Centroides vittatus, with the little scorplings that have now crawled out of the mother and onto her back. And they will stay there for the first molt, about a week. And then they've got to disperse because the mother might eat the babies, which is a really interesting question, too. Okay, so there's a little bit about pectins in use for mating purposes, which is awesome, right? And the males have larger pectins than the females. But females have pectins too. And really, to track a pheromone, you don't need such a complicated organ like the pectins are, because you just need like a snake's forked tongue to go right or left to be able to track your way to the female. So we think there's more. We do. And this is what's most exciting to me. And the title of my talk is Navigation by Deja Vu. But more appropriately would be navigation by déjà goûté or déjà touché, by taste or touch, sort of like the sense that you've tasted it or touched it before, and you can get back to where you were. Okay, so let me try to take you through this, and then we'll clean it up on the backside. But these scorpions that I study live in these beautiful sand dunes all throughout the Southwest. This happens to be in New Mexico, well, in a place called Sevieta. It's just gorgeous. And they have burrows. If you get out of your car and look around, you'll see burrows all over the place. Here's one. That's a scorpion burrow, little slit-shaped burrows. And these sand scorpions, what they do is they come out at night, as we said. They may come out to hunt. They may be mating, but that's usually once a year. But most nights, it's like this. They come out, they hunt, they may get lucky or not, but they got to get back to their burrow. And for me, this is the most interesting question. How do they do that? How does this animal figure out how to get back to its burrow? And so I want to give you three ideas here. I talk about the pectins and this idea called familiarity. Um, and you may have seen a paper I sent to Meredith that is in review right now that talks about this idea of navigation by familiarity. But then I want to show you how we've been trying to simulate this with some of our computer programs. So let's start again with the pectins. Incredible animals and organs. Let me show you some pictures of them in just a second. But first, the basic premise of this navigation by familiarity idea, it came out of the ant world and the bee world, mostly the ant, the desert ant world. It really hit the presses not that long ago, like about 2012 or so. And it really comes down to this. It's a really simple idea. To me, it's a really provocative idea. Um, how do you get home? If you're a bee or an ant or maybe a scorpion, and say you're an ant or a bee that's gone miles or kilometers away, how do you get back to your hive? You've got a brain the size of a speck of pepper. How do you do this? Well, this idea comes down to some really basic rules. One, look, taste, or feel around. If you see, taste, or feel something familiar, fly forward or walk forward. If not, taste and feel around some more and repeat. That's kind of the behavior rules that are implied here. And in bees and ants, we think they're using, well, strong evidence is that they're using their ommatidia in their really beautiful compound eyes. And we're thinking scorpions might be doing something similar, but with these organs, instead of looking out, that they're tasting and feeling straight down. So let me just zoom in on the pectins then. And maybe you, when you knit your socks, you can somehow include these pectins in there because they are one of the most spectacular taste organs on the planet. So they hang down from the mid-ventrally from all scorpions. Um, they're composed of a couple of flexible spines, if you will, and a series of movable teeth. And these number of teeth vary depending on the animal. By the way, here's the genital opening I mentioned before. Um, but if we get really small and look down in a scanning electron microscope, you see on the tips of each of the teeth are hundreds, and in some cases thousands, of microscopic dots called peg sensilla. Let me blow this up further, just blowing up a little patch. Let's just say, let's take a little patch of these peg sensilla. And now we're down in the 5 micron range, so you can never see this with your naked eye. But you see these structures called peg sensilla, and they're all lined up in the same orientation. They emerge from a flexible socket, so they've got this flexible cuticle, so they can bend. And here is one of them. We're looking right down the throat of one of these. So here's the flexible cuticle, but right here is a terminal slit. There's an opening that allows chemicals to get in. And I don't know if I'm in your way here. I'm not sure. I'm moving myself over to the side here. This is a cartoon I drew that shows what's going on inside. Basically, in every one of these tens of thousands of peg sensilla, there is a population of neurons. And there's about 10 in each. And of those 10, there's one, which I've colored in blue, that is a mechanoreceptor. It responds when this peg is bent. And then the other nine respond to chemicals. So most of what this thing is a taste organ. It's a tongue. This is a tongue. It's a gustatory organ. It's a taste touch organ, just like your tongue is a taste touch organ. But this might be the most complex tongue on the planet, really. It's just phenomenal. And we can do things. For example, right here, I'm sitting next to what's called an electrophysiology rig. Well, what we do is we would put these animals under the microscope. So you cool them down and you carefully display them with their pectins elevated towards the lens. And then we make little tiny electrodes out of tungsten. We electrolytically carve these things down. Sorry, I'm just moving some things here. Yeah. So we carve these electrodes down to a tip that's less than a micron. And then using a very sensitive micromanipulator, you can then maneuver this electrode through through that flexible cuticle. And then you have to amplify it about 10,000 times, and you can play it through a loudspeaker and visualize it on the oscilloscope. This is what it looks like in the lab right here. I've got a big, powerful microscope on a vibration-free table. Here's the manipulator. There's a scorpion down here. There's an oscilloscope and some electronics and so forth. But what I'm going to do is play for you what it sounds like when you make a recording. Here we go. This is a peg sensillum. I just poked in, and you're hearing action potentials, neural impulses from those neurons in the peg sensillum. In a second, I'm going to blow a 1 second pulse of hexanol right here. So I just blew a 1 second pulse of pure hexanol across the tip, across that little slit of the peg, and you see the response. There it is. The action potentials get faster, right? They speed up. And that shows me this is a very strong, good response to hexanol. And there's even other cells. I won't go into this, but there's cells that are inhibitory that kick in as well. And we've found through the years that these organs respond to all kinds of things. They respond to aldehydes and ketones and esters and aromatics. And some are excited like this, in other cases, they're inhibited. So we just got this really incredible repertoire
Hi,
of
Doug.
response.
Sorry,
Yeah.
excuse me. It doesn't seem like we're getting any audio from your recording. Were we supposed to be hearing something? I checked the host tools, and you have permission to share on my end.
Yeah. Is it is it now? Hello?
No.
Oh, stop share. You're saying you can't hear me?
Yeah, we couldn't. I can hear you fine, but I'm not sure if we're supposed to be hearing an audio from your PowerPoint. I couldn't hear the Al Green earlier.
Oh, okay. Well, yeah, maybe that's just the nature of the beast
All
here,
right.
unfortunately.
Well, sorry. Just wanted to check, but thank you.
Yeah, the good news is I don't think I have any other audio going on. So anyway, so what you would have been hearing is pops. These action potentials, we play them through a loudspeaker, and each one of these lines will sound like rain. I mean, it sounds like pop, 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 pop. So it's really kind of cool science if you've ever done this. So anyway, as I said, we've found that these pegs respond to lots of chemicals, but they respond similarly. So all these pegs, basically, they may respond to alcohols and aldehydes and ketones and esters, but by and large, they all respond the same. So this is wild. You've got this redundant set of pegs. I mean, what is up with this? Tens of thousands of these units with these repeated chemoreceptors. And so we've done some math. I mean, the the actual sort of resolvability is in the trillions to gazillions of numbers I can't even pronounce of that is different patterns that these organs, if you press them to a surface, the potential number of patterns that these things could detect is just astronomical. To me, it's like QR codes. These QR codes are just full of information just with these squares of black and white. So it's this matrix power that we're thinking about. And just to kind of bring this home, you may be saying, okay, I kind of get it. Maybe if you've been down a path and you are able to memorize all these tastes and touches that lead you back to your home, your burrow, then maybe all you got to do is kind of look around and go towards whatever is most similar, whatever patterns are most similar, and that will take you back to where you were. And that's great. That will work, but there's an inherent problem here, and that is when you leave your burrow, everything is 180 wrong from when you're coming back to your burrow. So if you're leaving your burrow, like say this scorpion here, and let's say just for giggles, the blue circles are on the one side and the triangles or the diamonds on the other. Well, when the animal's coming back, they're on the wrong pectins. It's like everything would be 180 wrong. So the patterns just wouldn't make sense. So this is a big conundrum, and well, ants and bees have solved it, and the way they do it is by path integration. And if you may know that if you've raised bees, before a worker honeybee goes out for her flower, for her first time out, she backs away from the hive and swoops around the hive while facing the hive, and then goes back to the hive. And then the next time, she goes a little further, all the while facing her hive. And what we think she's doing is acquiring these matrices of familiar scenes that would lead her back to her hive if she ever sort of falls into that area again. And it turns out these desert ants do the same thing. They actually, first time out, they do these learning walks around their nest, their burrow, their nest, before they go out and find their first food source. They do these learning walks. And so we thought maybe scorpions do learning walks. Is it possible they do this too? And and that's called path integration, really. That is, they don't just follow their breadcrumbs back or some pheromone back. They take essentially a hypotenuse back. And the way bees and ants do it is they use vectors, these things called E vectors in the sky as a global reference to update their twists and turns. And ants actually count footsteps to know how far they are. So they get an approximate measure as to how far to go back. Well, we think scorpions might be doing the same thing. That is, if they are able to get back the first time using path integration, then they would acquire those scenes, and all they got to do the next time is just follow what's most familiar, and they'd get home. So we went looking for this question: do scorpions have learning walks? And it turns out they do. So over in Sevietta, we made this simple system where we made a water heater pan with some sand and we made a mound in the middle and we tried to induce the scorpions to dig their own burrows. And then we tracked them all night long. And this is what it looks like in the lab. It's just, we just filmed them all night to see what they did. We put a little bling on their back, a little crystal to improve the resolution so we could see them. And this is an all-night filming, for example, of this is the mound, and this is one animal that was caught every second all night long. And you see they're very active, but they will dig burrows, which is cool. Here's one caught in the action of digging a burrow in the mound. And what we were interested in is what do they do in the very first seconds after they dig the burrow? And here's what we found. And what you're going to see is a time-lapse video of an animal that's about to dig its burrow for the first time. This is sped up about 10 times, but the white dot is the scorpion, and right soon it's going to move to the middle and it's going to dig its burrow right there. That's its first digging, and now what happens is it does these loops. And this is terribly exciting to me because oh my goodness, for this familiarity idea to hold any weight, there's got to be a way to get your initial homebound glimpses, if you will. And it turns out every single animal did it. Everyone we film, as soon as they dig the first time, they start doing these loops. They loop out and come back. They loop out and come back. They loop out, come back. And uh, and in fact, like this one here did its first set at time zero, and then 10 minutes later did some more, and at 25 minutes later it did some more, and at 47 minutes it just went crazy, going out and back, out and back, out and back, and out back. So we think this is evidence for learning walks in scorpions, and we think it might be sort of what's necessary, this innate behavior to get goal-directed glimpses. And so in the paper that you probably is kind of a real snoozer to try to read, we looked more closely at this. In particular, like this one right there. This is one animal that did some really cool behavior, and I'm going to blow it up here. So here's its all-night filming. But what we found was that initially, after it dug its burrow, initially, it did these loops, these what we're calling learning walks that goes out and back, out and back, and out and back. And then a little bit later, a couple of hours later, it tended to pick a consistent inbound path that we call directed movements. This is crazy. This so it's like it's kind of choosing one of these spokes, and then it over and over and over it uses that to get back to its burrow. But it doesn't go out the same way. It goes out any number of ways to go back to the wall, but then it kept going towards the wall along a consistent path. And so we're thinking, oh my goodness, this is kind of like what the bees and the ants are doing after they've done their initial learning walks or learning flights. So this is where we're at. We've modeled this behavior in Python, and I'll do a really quick once through, and then we'll open it up for questions. I'll just show you what we've been able to accomplish. We've tried to make a model scorpion based on all the information that we've gleaned to see if this is even plausible. It actually came, this is a much younger version of me. This is my brother Arthur, who's the real genius. And he had this idea back in the early 80s where he thought he knew how bees and ants were navigating. And he built this little thing. He built this artificial bee, if you will. So this is to emulate their little compound eyes. And it's got a processor and it's got some memory. This is from 1980s, but it actually works. And I know you're not going to hear the sound, I guess, but what I'm actually going to be doing here is running this wand that he built over a black and white landscape, just a simplified kind of sand-like landscape. And it's acquiring these matrices. It's only 16 or 25 photodiodes, but it's saving those to memory. And so to then recapitulate, all you've got to do, you can close your eyes and you listen to it. And I know you can't hear it, I guess, but it's making these buzzing sounds every time it detects a matrix that is similar to one it's seen before. And so you use it like a Geiger counter, and you can find your way back to where you were. Just using those simple rules of look around, step forward if you see something familiar. Otherwise, look around some more and repeat. And we then put this on a cart, and this is my assistant, Brad. We put it on a little cart, and we used vision instead of looking straight down. It's a little camera. And here comes Brad, and he just made this some circuitous route through the hallway of our building. And then he gave me the cart. I was sequestered away, so I didn't see what he was doing. He just gave me the cart, started me back where we started, and I am to try to guide the cart based on the familiarity. It's actually it's both popping and it's also lighting up with a stronger dot, if you will, the better the match. So I don't actually see the images. I just see this dot that's either light or dim. And my goal is to try to make the dot as light as I can. And watch what happens. Here I come. And I can retrace the path that Brad took. I almost hit that guy. Sorry, right there. And so it's this simple idea called familiarity, and it's like I liken it to like a water slide. Like if you get in this catchment area, you just can't help but to go to the end. You know, it's like in a water slide, you start in the top, and it'll take you all the way to the end. I don't know if this is making any sense at all, but we've done a lot of modeling of this. So here's my latest version that's in that paper that you have, just trying to show the premise of it. And if you really want to understand this premise, I would watch this video over and over about 1,000 times because it's showing as simply as I can show it. Um, what you're seeing is a little training route being formed. These are the pectins in memory, that basically the glimpses in this circuitous route. And then you start it near the beginning and it looks around and it looks at the current glimpse compared to memory, and it saccades left and right. And whichever is the best match, you step in that direction and you repeat. I'm going to do it one more time. And this is the blow up of it down here. And this graph is basically showing you the best match and the step that the agent has to take. And look what happens. It gets back to where it was, just using that simple set of rules. And so we've modeled this. A former student of mine named Albie made a beautiful example of this. So it's going across this black and white landscape. It's showing the pectins up here in the upper right. Um, and then you can even do this with a very simplified version of the pectins. And this is cool because it almost got off track, but then it latched on and it easily makes it all the way to the end here. Let me do that one more time here. And you can see it. It's just, these are to emulate the teeth of the pectins and in just a black and white world. So a terribly simplified world, which we know is way too simple because we know these pectins can pick up on hundreds of chemicals and textures and such. But this at least shows proof of concept that it is possible. So we're right now trying to make a robot that does this automatically to navigate across terrazzo floors. And then I'll leave you with this one. This is an actual simulation that is as big as a scorpion home range. So scorpions will go out, a meter or two and get back to their burrows. So I just wanted to see if using this simple premise of going over, you know, that is making an initial journey that's very complex and putting the animal back at the beginning and then just having it look around and move towards whatever is most familiar in this matrix by matrix sense. Can it make it all the way back to its burrow? And so I'll just let it run. You're seeing three things in the bottom. That's because it's both looking left and right as well. But the green one is the one to maybe track. And you can see the purple is the actual animal very accurately. It actually got off path right there. Look at that. And it moved around, meandered, and it got lucky to catch the path again. And it went all the way to the end. And something cool happens right over here. When it gets to the end, it does a prediction that's completely in line with what we expect. It goes around in circles because it's lost. It doesn't have any more familiar scenes. And so it's just now saccading. It's basically turning in circles, trying, and, look, basically its whole algorithm is always move towards the most familiar scene. And since nothing's familiar there, it just ends up spinning around in circles. It's saying it's like the burrow's got to be here someplace. Well, that's enough. I know that's too much, but I hope you agree that lesson number five is true. Pectins are really cool. I'm going to thank all these people. Oh man, I've had so many students over the years and so many sources of funding, and I'll just ask for any questions. Thank you so much.
Thank you so much. That was absolutely incredible. I think my advisor is going to be really annoyed when I go in for our meeting tomorrow and say, I want to study scorpions instead now.
Good. You. I hope you do.
Well, we are collecting. Feel free to drop questions in the chat, but while we're collecting our thoughts and figuring out how to ask all the questions we have, would you mind just taking a couple minutes and telling us why you love what you do beyond lesson five, why pectines are the coolest ever? Why do you love what you do, and how did you get into doing it?
Yeah, that's a great question. I love that question. Um, it really was my brother that I used to teach high school in the Middletown High School. I taught in Northern California. I was the science teacher and the basketball coach, and I was a pretty good teacher, but I was a terrible coach. Anyway, at that time, my brother built that thing that you saw, and he got a little patent on it. He was just watching Yellow Jackets one day fly, and he had this aha moment and said, "I think I understand how they might be getting back." And he then built this model and showed sort of proof of concept that this might be possible. Um, but by that time, the bee and ant literature had not stumbled on this idea of familiarity. I'm telling you, it didn't really happen until about 2010 or so. In my opinion, it came out of the University of Sussex, a group there. So I thought it was the coolest thing I ever thought about this idea. How can this thing, this bee with a pepper brain, go miles and back to her hive and get back to that flower and never get lost? Better than the best drone we can make. There's no drone that we have that can outperform a bee. And I was hooked. We didn't call it familiarity. We called it sequence pairing. But I quit my high school teaching job. I just knew I had to go back to grad school, and I didn't do anything right. Uh, I don't know. Don't follow any of my advice, but somehow it worked out. I quit my job and I rode my bike to Canada. So I was in San Francisco area. I was just going to stop at grad schools along the way and say, "Hey, I got to study bees. This is what I want to do." And they looked at me crazy. So I would just knock on doors, looking for advisors. You're not supposed to do that. Okay. But I met this guy named Phil Brownell. You may have seen his name a couple of times. I met him up at Oregon State University, and I told him he listened. I had this long hair. I looked like a crazy guy coming in on my bicycle, and he listened, and he said, "Well, that's cool. Bees are cool, and not much is known about their navigation. That's true." But he said, "Have you ever seen this animal?" And he went in the back room, and he came out with a scorpion, just like holding a scorpion and a black light. And I said, "Oh my gosh, I'd never seen a scorpion before." And he said, "Well, bees and ants are cool, but there's hundreds of people studying bees and ants. There's no one studying this animal. And besides, they've got this organ on their bellies, right? You know, they got this organ down there called a pectin that no one knows anything about." And so that's my story. I said, "Well, if you'll have me, I'm willing to try." And I had to take my GREs and all that stuff, and I got in. I got lucky to get in, and so I've just been studying this organ and kind of creeping up on this idea of navigation. And I think we've now got enough background information about these pectins to start to tell this story. It's far from over. It's got so much more to go, but that's my story. And I really don't feel like I've worked a day in my life because I'm so passionate about this. We would stay up all night studying these animals. It's just such a pleasure when you are on to something. You just don't feel like it's work.
That's an incredible story. Thank you so much. I think that a lot of us are familiar with feeling that kind of excitement and passion about our research projects. And it was so great hearing you ride your bike into these different grad schools. I can't, I think the current, the contemporary of that would just be, the millions of cold emails that that you send the very nicely worded door knocks.
Yeah.
But okay, looks like we have some questions in the chat. Oh, I also wanted to mention I worked at Sevietta for my first two field seasons. I worked
Oh,
there on ants and aphids and yucca, and that's where I fell in love with desert ecology. So I did my master's in Philly, and after two field seasons at Sevietta, I was like, I have to be in the desert. And I only applied to PhD programs in Arizona, Sonoran Desert, Chihuahuan Desert, Mojave Desert. It's such a special place. It's so
it
wonderful.
is. What a field station that is, right?
Oh
So
yeah,
gorgeous.
it's gorgeous, and they just got a new director who's doing some really cool stuff. Sorry, I'm getting super off topic, but okay. Our first question from Bryn is: are there any exceptions to internal fertilization and spermatophores, or do all scorpions use this mode?
Uh, that I think all do. I don't think there are any external fertilized animals.
Do I have a little bit of a follow up to this? So, do female scorpions? I'm kind of thinking of odonates here, but do female scorpions possess any, adaptations towards removing the spermatophore once it's been deposited? Once she's been dragged over it, does she exhibit any sort of partner choice in that?
Yeah, it's a great question. Um, I'm not well versed in that. I don't know. Uh, I'd have to send you to some of my colleagues who are very much into reproductive ecology, but you would think that that spermatophore would preclude other males, right? And so
Yeah.
I don't know if she's able to extract it or if something wasn't quite right in courtship or what.
Now, I sent out a Google form over my newsletter, and so there are a few people who weren't able to join us today, but did want me to ask you some questions because I'm recording this talk. So, people who read your paper, just scorpion enthusiasts,
Mhm.
and I'm going to try to add those questions in when there's something in the chat that's kind of a logical progression. So,
Sure.
I have a chat. I have two questions, kind of. One of them is how many times will a female reproduce in her life cycle if they have these 12-year pregnancies. How many times is she going to reproduce in her life cycle?
Right.
And then, I guess, how can you tell males from females?
Right. Yeah, so 12-month pregnancy gestation period. Yeah, it depends on the species. The ones that I have here, the Utahensis, they will live several years, maybe up to 5, and they can mate multiple times. So it's usually in the second year that they would be sexually mature enough to mate. And so it adds to some of the questions because these are cannibals. So why would the mother eat her own babies? And it could be a tie to the environmental conditions. If you think about it, that say we have a very poor vegetation year, which means a poor cricket year, which means little food for the scorpion, yet the mother is giving birth to these babies, it might behoove her to eat her own babies and bring resources back into her so she can get through to the next year. So there's probably a very interesting evolutionary study, selection study right there. It's probably not been followed up much in the literature, but man, oh man, there's some really interesting questions there. The males and the females in some species, especially the ones that I study, are dimorphic. The pectins are dimorphic. So the males of the Utahensis will have like 30 teeth on each of the pectins, and the females have about 20 teeth. So the males, you can see them. As I look down on the male, you can see that his pecten extend a little bit beyond his body, and the females are quite a bit smaller. But the females have pectins too, and they have thousands and thousands of pegs and stillas. So this has always been my problem. It's not just about the males finding the females. We're thinking there's more to it, and so we've stumbled on this idea that both males and females are using them to get back to their burrows.
Incredible. Thank
Mm.
you. Our next question is from Jillian, who has two questions. So, number one, with the familiarity walks and the size of the scorpions, how significant of a change to the environmental surrounding the burrow would be needed for them to become lost? And
Yeah,
then, second, are scorpions attached to maintaining their burrows, or will they abandon the old and create a new?
great. I love this stuff. Um, yeah, in that paper I sent you guys, which is under review, it's in a viral archive right now. I think it's about figure 5 or 6, where we did a study using our model, where we systematically altered the sand, if you will, of the landscape. We both randomly changed the pixels and also the size of a gap. So as they're trying to as they're navigating back, we confronted the agent with disrupted sand, if you will. And we were able to bracket this and figure out that, well, if you've got a narrow gap and not much disruption, they can cross it without much trouble because you've got inertia in your favor, and it's even if it's changed a little bit, it's still going to be the most familiar compared to a completely different scene. But there comes a point if you make the gap too wide or if you disrupt the sand too much, then yeah, they will absolutely get lost. And but in that last one that you saw, I actually built into that one an algorithm that if you are off path, that is, if your match is not that good, then turn more left and right and take shorter steps. And that's why that one was able to regain its path, if you will. And we see this in animals, we see it in ants, we see it in bees. That is, if they come to an unfamiliar spot, they start doing these expanding spiral meanders, if you will, which is a very clever, I say clever, a very beautiful selected behavior, right? If you are in an area that you've lost your path, well, if you expand out, you're likely to catch another one that's going to lead back to your home, to your burrow. So we think they do that. I've actually got a video that shows some evidence of that. I don't have that published. But I also will not dismiss their eyes. As I mentioned, their eyes are terribly understudied. If you look at some of the early accounts of scorpions, they all say that scorpions are poorly visual. I even said that. You can find several of my papers that say scorpions are poorly visual. That is not true. There was a guy named Fleissner, well, a group Fleissner and Fleissner did some measures of these medial eyes, and they are really sensitive. As I said, they can see in starlight. I am sure they can see bushes, their whole panorama in starlight. So you've got that too. So if you're off, I think they're doing multimodal stuff. I've just been focusing on these pectins, but I would certainly think they're going to use every trick in the book, use their vision, use their pectins, whatever. They've been around 400 million years, so there's been a lot of time to really sculpt natural selection here. You're still muted. Sorry, Meredith. I think yeah.
Okay, thank you. I think the second part of that question was just, are scorpions attached to their burrows, or will they abandon and create new ones?
Yeah, so it's different for each species. The ones I'm really enamored with are these sand scorpions that tend to be faithful to their burrows. Females more so. The females are really quite faithful to their burrows. The males a little bit more. They move around a little bit more, and they may dig new burrows. Although they certainly show a degree of fidelity too. But as I said, it's the males that pack up and start wandering. usually in August and September, tracking down females, and after which they'll have to dig their own new burrows again. But by and large, these females are really quite faithful. There's been a lot of work out of the Gary Paulus, who's a famous scorpion biologist that died tragically in the Gulf. But he's did a lot of really cool ecology studies of scorpion movements and found especially the females are really quite faithful to their burrows.
Incredible. Thank you. Our next question is from E, who asks, I'm interested to hear more about your Python Scorpion model. I program in Python. Is it available anywhere?
Yeah, great. Man, I would, yeah, it's, Python and AI these days, right? Um, if this thing gets published, which I really hope it does, then I'll make it freely available to everyone. But I can send Eve, if you want me to, I can send you a bare bones version of it. So, which is probably the best way to learn it. It's kind of that you saw that simple one I went over a few times that showed the visualization of it. I'd be happy to share that with you. I'm not sure if you can, Meredith, I can send you the Python code and you can distribute it. And then I would love, Eve, if you, is it E or Eve? I'm not sure what your name is.
Elizabeth.
Elizabeth, if you can try running it and see if it works on your system, I'd love to know. And in fact, I'd love to know if you see any efficiencies, ways to make it even better.
I
It's a
can
really,
definitely act as a point of contact there. I can also drop your email, Dr. Gaffin. If you're okay with me sharing the ou.edu address, I can drop your email in the chat right now for anyone who might want to ask further questions or get a hold of some more code.
yeah, let's do that. That's great. Happy to do it. I really, I mean, to be honest, I'm an old guy now, and I want my pizza being delivered by this method. You know, there's there's no way, there's no reason that these self-driving vehicles should not be using this familiarity algorithm. It is so elegant and so simple. It doesn't require fleets of satellites and so forth.
Well, Elizabeth, please update when you run through that if you check it out. I'm curious to know where that goes. Then our next question is from Maria, who says, did I understand correctly that scorpions explore on their way out but develop a preferred route home at the end of the night? Is there any insight into why they might use the same routes back to their burrow? That's the first part.
Yeah, it's the opposite. We think that they um, before they go out on their longer range hunting excursions, they do these learning walks. And I've seen it. You can see it too if you're patient. Early in the evening, just when the sun's going down, and you get your black light out, um, you can see usually scorpions at the threshold of their burrows, and they're just getting fixing to come out. And so if you just watch one for a while, once the sun goes a little bit more, now they are bugged by the UV, so you kind of want to play a game by not looking, don't shine it right on them. Try to just have a little dim light, or if you have an infrared camera, set it over the top and you can capture it. And we think we see them. And in fact, I see them sometimes when I'm picking up a scorpion. I see it in the side view, another one doing these learning walks. So we think they're laying down these familiar paths early in the evening, and then they go further afield. They don't go just willy-nilly, they just don't go they don't go that far, a couple of meters. But usually they're drawn by something scurrying on the sand, like a cricket, and they will chase it. Again, using the slits and scylla, the vibrations, and sometimes they catch the cricket, sometimes they don't. But if either way, they got to get back. And that's where we think they probably use path integration, that they have some measure of the angle and distance to their burrow, but it's not precise. It's approximate information. And there was paper out of Germany that showed another version of path integration in scorpions. But it'll get them in the right direction, and then they'll stumble on these pre-laid down learning walks, and all they got to do is pick one of those because they'll say, "Oh, got it, got it. I've seen this before. I've tasted or touched this before," and it'll take them back. Meanwhile, they've added a new twig. You see, they've added this additional twig, and they've even made this path wider because it's not precise. They just kind of they'll go back and forth across it, and you may have even seen it in that behavior example, how it got wider and wider. It just becomes a freeway almost of what looks to be familiar information. So that's what we think is actually going on. And we think they start over every day, like spiders building new webs every night because the sand wipes clean. It's hot out there and it's windy. And if you go out there during midday, everything's wiped clean. So we think that they start anew because down, you might say, well, the things are just going to blow away at night. It's actually in the swales of these dunes. If you go out there, it turns out in the evening it's really quite calm in these swales and the wind dies down. And I've taken pictures early in the morning, just as the sun's coming up. You can see every single little footstep of every little bug that walked across the sand by just the shadows, if you will, as the sun's coming up. So you can see that that sand is really undisturbed. It's amazingly well preserved through the evening, but then all bets are off because the winds come and the sun comes up and the heat and the same picture, all those footsteps are gone. Every little thing is gone. So I think that they start anew. I think they're actually building these recognition pathways every single night.
Would the type of environment that they're in determine that? I mean, a sand scorpion versus a scorpion that's living in more of like a leaf litter kind of area, would that, do you think, affect their memory?
Mhm. Yeah, great question. Oh my, I'll give you another. Well, I hope some of you take me up on some of these projects. These sand scorpions are special because they are faithful to their burrows. I think like your Centroides, Explicator sculpturatus down there, I don't think they're as faithful, but there's much less literature known about them. They may have some site fidelity, but they're often, tolerant of each other. They're not so cannibalistic. You can sometimes pick up a piece of bark and see hundreds of them under there, right? Um, but I think that so it's going to vary by scorpion, by species. But the ones I would love to do are some of my friends tell me there are these hadrus, the hadrus are the big giant hairy scorpions, that there's some of these on the rock walls around Moab, that they live basically their lives on the sides of these rocks. And that would be fantastic to see if those guys are going back to a common retreat. Tag them and just put either radio, you probably put a radio transmitter on them. They're so big. See if they're faithful to a spot. And then, oh my goodness, then you want to know, are they doing learning walks and are they taking precise pathways across this rock? What happens if you scratch the rock, Because sand, and maybe they don't have to start over. Maybe that tapestry is there for the long run, whereas these shifting sands are, renewing every single night. So I think I would love to just go on a camping trip with a camera and film some of those hadrus sometime.
That would be such an amazing project. And yeah, I just started doing mark and recapture for some of my ants. I think it would be a lot easier to mark a scorpion than a
Hmm.
tiny little ant.
Yeah.
Okay, I have another question from the Google form, which is, why do they glow green?
Yeah. Yeah, it's still an open question. Well, why? I mean, it's like how they glow green. There's a couple of chemicals in their cuticle. One's called beta carboline and the other's called coumarin. Um, beta carboline is actually in cataracts of like dog's eyes. Apparently, it's the same material. I don't know. I've never shined a black light in a poor dog's eye to see it with cataracts if it's glowing, but it's apparently a similar material. And then coumarin, I think, is related to a blood thinning agent. This is weird quirky, just probably weird, quirky quirks of biology, but those are the chemicals that have been identified. So what's happening is you shine ultraviolet light on these molecules and it excites these electrons to a higher energy state. And then when they fall back, they emit fluorescence, packets of fluorescence. So it's not autofluorescent, it requires the energy of the ultraviolet light to excite the electrons to a higher energy and falling back, giving off packets of fluorescence. So that's how they fluoresce. Why they fluoresce and why green? We think in that one paper I highlighted, we bring out some of the arguments, but their eyes are maximally sensitive to green. So these compound eyes, and secondarily in the UV, they've got two peaks. They've got a UV peak, which is smaller, and a big green peak, which is weird, right? So you've got these eyes that are maximally sensitive to green, but you've got these animals that fluoresce green under UV. So, and you got a secondary peak of UV sensitivity in the eyes. And so it's got us just thinking, I mean, it could be absolutely nothing. It could just be some byproduct. But um, we have strong evidence that these animals are sensitive to UV, and then this has actually been known from the 50s. In fact, there's even some obscure papers that suggest that there are tail sensitive elements to ultraviolet light. Okay, you can find a couple of these without much difficulty, and but so the thing that we did is that we set up an assay where we would test the animal's locomotory behavior under under low levels of green and low levels of UV. And we found that the animals responded similarly. We just did them in these little arenas and filmed from above with an infrared camera. But actually, I think we were filming from below through some plexiglass, but shining light from the top. Um, and we found that when we clicked on the UV or the green, they would get annoyed. They would run in spurts, if you will. But when we covered their eyes, we've made these little goggles and covered their eyes with foil. They they still would run under the UV, but not under the green. So it made us think, oh my goodness, their whole body might be detecting this UV and transforming it somehow to green. It's like green's the answer. It's like most of the photoreceptors seem to detect green, but I don't know. Now I'm hand waving. Is it like a fiber optic system through their cuticle? You saw how bright they are. I mean, they're just stunningly bright. They're the brightest thing out there. But again, the night sky, this is like the sun being on top of you. It's not like this at all. It's very low levels of UV. So it's still an open question, but we've got these tantalizing bits of information. We know they're sensitive to UV. And our best idea, you can do this too. When you're out there on the sands and looking for scorpions, just pay attention. You might be surprised to see how many times you find one scorpion under a little twig, just an isolated blade of grass or just a twig. And I often thought, "Well, how did that?" Because it's otherwise sand, one blade of grass and one scorpion under that. And I'm thinking, how did that scorpion find that blade of grass? And so it's kind of has taken us to this hypothesis that it probably just ran until any tip of it was shaded from whatever tingly UV feeling it's feeling. And by reflexive turning, it moved under the twig, probably escaping a predator like an owl or a mouse or something. But it's by no means a closed issue. This is still an open question, and we've just got a couple of little clues here. So I hope one of you will pick this one up too. There's so much to do. Oh my gosh, there's so much to do. And I'm telling you, this other hypothesis, I really want some. I can almost lay out the experiment for you. I would be shocked, kind of surprised if those males aren't tracking females using these amazing eyes, right? And as I said, maybe they're seeing them as black holes, right? That the energy is being absorbed into their body and not re-emitted. Only 2% of that UV energy is re-emitted as green. So 98% is being absorbed, but it's bouncing off everything around them, right? The sand is not absorbing it. So I is just dying for someone to do this test. Here's what you do. You go someplace like Sevietta in the mating season. Yeah. And get some infrared cameras and get some arenas. So find males and find females, but put a female in a clear arena at a little bit of distance, but film the male in his own arena and see if where you place her, does he orient? Is there any tendency for him to orient towards that female, right? Don't even put a black light out. Just let the natural night sky, whatever is out there, be the energy source. And just see after doing this over tens or hundreds of animals, when you do the math and do your circular statistics, is there a tendency for an animal to be directed towards the female, right? And then you can do all kinds of other things. You could paint her, to cover her fluorescence, or you could make artificially fluorescent animals and all kinds of things. But that's the first step. Because I'd be kind of shocked if a male with these great eyes and low light and seeing and that can see in green, but would see also in the UV, but maybe the absence. I just feel like they're seeing a silhouette. But it's natural selection. This guy's got to find a female and pass that information on. So I think that's a hypothesis that someone just has to test sometime.
Well, thank you so, so much for your time, for all the dissertation ideas.
Yeah.
Um, it's been so, so wonderful listening to this incredible presentation and learning more about these animals. Thank you so much for sacrificing part of your Sunday afternoon to giving a talk to a knitting circle.
Oh, this is so much fun. Can someone knit some little pectins on your socks? Is
I
that good?
will. I was actually, if you would like, I mentioned this in our initial email, but I would love to send you a pair of socks or hat or something that has a knit scorpion on it. And if you would like that, just let me know where I can mail it to, and I will absolutely add pectins on there.
Oh, I would
They're
love that.
they're a chart of about what I forget the dimensions, like 10 by 15 pixels or something. So there's not a lot of detail that can be added to it, but for a knitting pattern, when you publish it, it's kind of like a recipe for a sweater. And when you read a food blog and there's all the information that you don't really care about at the beginning of the recipe, you scroll
Hmm.
forward. Well, hopefully people aren't scrolling through with this, but whenever I send out a pattern, it includes a page and a half of information about the organism that's depicted in the pattern. And in that information, I do say very clearly, here's the diagram of the scorpion, here's the correct number of legs, here are the pectins. So people will know.
I love it.
Well, yeah, thank you so, so much again. We really appreciate it, and I hope you have a wonderful rest of your weekend. Thank you so much.
Thank you so much. Appreciate it.
Bye,
Take care.
everyone.
Bye bye.